Hey guys, welcome back. This time around we're going to take a look at the histology of the nervous system. Now remember, the term histology is to microscopically view the tissue. If we were microscopically viewing the nervous tissue, we would find that it is about 80% cellular, highly cellular in content. It is tightly packed and intertwined. The nervous system is going to be composed of two types of cells, the support cells and the neuron. Now, when we're taking a look at the support cells, we will find that the support cells are going to be small. They're going to be non-excitable, which means they do not conduct any electrical events down them. And they're going to be important in surrounding and supporting the neurons, the other type of cell that we would find in nervous tissue. In general, they all provide scaffolding, segregation, and insulation for the neuron. These cells collectively are called neuroglial or glial cells. They have profuse branching, vastly outnumber neurons, and account for most of the firmness of the nervous tissue. There are 6 types of glial cells. 4 of the glial cells are going to be found in the central nervous system, and the last 2 that we'll discuss here, are going to be found in the peripheral nervous system. It'll be important that you know these different support cells and you know their role in the overall health of the neuron. The first that we take a look at is going to be astrocytes. Now, astrocytes are going to be the most abundant of the support cells. They're going to be star-shaped. They're going to be accounting for about half the neural tissue. They have numerous radiating projections with bulbous ends that cling to the neurons and to the capillary beds, anchoring the neurons to its nutrient supply lines. They play critical roles in the immune response because they act like antigenic presenting cells. They also regulate the ionic environment in the extracellular space by minimizing the loss of potassium during repolarization events. And they recapture and recycle the neurotransmitters that are being broken down at the postsynaptic end. The second type of support cell that we would find is going to be a microglial cell. Microglial cells are going to be small cells that are going to be oval-shaped cells with thorny projections. And they function in the central nervous system as a macrophage. Remember what a macrophage is? A macrophage is a cell that's specialized in eating microorganisms and or cleaning up the cellular debris that may be accumulating there. They're sort of like the garbage collectors of the central nervous system. Epidermal cells are going to be the third type of support cell that we have, and they're going to be anywhere from squamous to columnar-shaped cells with cilia that is going to project out into the central cavity. Now, these epidermal cells are going to line the central cavities of the ventricles that are going to be found in the brain and the spinal cord. Now, the fluid found in these cavities is going to be the cerebral spinal fluid. Now, these epidermal cell lining is going to form an impermeable fluid-blood barrier, preventing things that's in blood from migrating into the cerebral spinal fluid. This is the barrier between us and our brain and any of the microorganisms that may invade our blood. Now notice that these are specialized to have cilia that are going to be found on the central cavity surface. Now the cilia, if you recall, is going to be hair-like projections that work in a wave kind of formation against gravity. Now imagine as we stand or sit upright, the cerebral spinal fluid that's found in the cavity is going to want to migrate via gravity down to the lower tip or the inferior tip of the spinal cord. It is the responsibility of the cilia to ensure that when that gravity wants to pull the fluid down, it starts working against gravity to ensure that the ventricles in the brain are kept bathed with the cerebral spinal fluid, regardless of what orientation our body is in. The fourth type of support cell that's found in the central nervous system is called the oligodendrocytes. The oligodendrocyte means few branches. They line up along the thicker neuronal axons of the central nervous system, and they wrap their cytoplasmic sheaths tightly around these axons, producing a whitish protein lipid-based sheath that acts as insulating covering. This sheath is known as myelin sheath. And wherever myelin sheath is at, we will see a quicker electrical event occur. This will be the white matter, if you want to think of it, in the brain. The two types of cells, support cells, that would be found in the peripheral nervous system would be our Schwann cells. Our Schwann cells act like oligodendrocytes. They insulate by forming myelin sheaths around very large peripheral nervous system axons. The more myelin's there, the quicker the conduction of the electrical event. But they also function as the macrophage for the peripheral nervous system, digesting cell debris. Now, unlike the oligodendrocytes, though, the Schwann cells are capable of regenerating themselves because they're dedicated to a single neuron. This is an important distinction here, because if we have a peripheral nerve damage to occur, it is going to be the Schwann cells that allow us to regenerate that axon, because the Schwann cell will act like a tube-like structure, providing a pathway for the neuron to regenerate itself or grow back into. Take a look at this image. You can see that the Schwann cells do not overlap one another. There are spaces in between them. Those spaces we'll see are known as the nodes of Revere. Sixth type of support cell is going to be the satellite cells. The satellite cells are going to surround a neuron cell body. Now, the cell body is typically going to be found in the central nervous system, but when it's found outside the central nervous system, the cell bodies typically congregate or cluster together. And that clustering together is called the ganglia. These satellite cells are thought to control the chemical environments of the axons. Next time around, we'll take a look at the histology of the neurons. You guys have a great day. See ya.